All right, so for today's lab, we're going to go over data displays, and this was not a chapter in your lab manual, so you guys didn't have to like read any chapter before your lab manual, but you likely went over this with Johnson in lecture, hopefully sometime this week. It is chapter 2 in your textbook that you're doing. So data displays are kind of like the first step of statistical analyses. We can't really look, usually data sets are like huge and they're like so many rows, and you can't really get an idea of what your data or how your data is behaving, what kind of patterns you're seeing if you're just looking at the numbers, right? So what we like to do is to try to graph our data so that we can actually see how our data is actually behaving. Now, the issue is that you have to use different graphs for different data sets. So it's like, you guys are probably all familiar with a bar graph, but you can't use a bar graph for every single piece of, or every single data set that you have. It just wouldn't make sense. And we'll kind of go through those examples today. All right, so a little bit of a review from last week. We have different types of variables, and these variables can actually help us decide what sorts of graphs we're going to use. So we have categorical data, which is like attribute or nominal. This is like literally categories, so there's no numbers involved. And so, it can be considered like a characteristics. We have color, sex, age, class. That was like our pipefish example last week where we had, you could do male or female or brown and green, right? So these are some sort of characteristic. It's not something that's measured, it's just a characteristic that is recorded. It's a category. Now, typically when we have categories, that kind of hints to us that you should probably do a box plot or a bar plot, and we'll kind of talk about that. Then we have numerical data, and it kind of depends on how many types of numerical variables you have. So for example, like if I had categories, I probably only have one numerical set of data. And that's like, for example, let's say we're measuring length of those pipefish, and we were recording male and female and green and brown. So I now have like two different categories, male, female, green and brown, but I have one numerical variable that's describing those this population, which is their length. So the length is a numerical variable. That's like your response variable that's going to go on the y-axis on your bar graph. So, but for example, like we could also have like two sets of numerical variables, and that would maybe result in like a scatter plot. So for example, if I'm looking at oxygen consumption or how oxygen consumption responds to change in temperature. Temperature is a numerical variable, so that is like you can have 20 degrees Celsius, 21, 22, 23, 24, right? It's a numerical variable. But then we also have oxygen consumption. It's like 23 milligrams of oxygen per second or whatever, right? All right, so if we have categorical variable, first and foremost, this is kind of what our data sets would look like. So some of these data sets have multiple categories. Some of them have just basically like one, or I should say factor. We have one factor and two factors really. So there's different types of data sets that we can see that have categorical variables. So this one would have like count data. So our numbers are actually discrete variables. So this would mean that we're literally counting the number of females that are colorblind, males that are colorblind, females that are carriers, males that are carriers, females that are not colorblind, males that are not colorblind, right? So these are all just representing counts. Like you counted 5 females that are colorblind, right? So those are all just specifically count data, but the best way that we probably want to visualize this data is through a bar graph, because we do have these categories. And so the best way to kind of separate those and visualize how this data is set out is through a bar graph or some sort of bar graph. Now we can also have these different sorts of variables where you're analyzing means. And so you have these categories that have different, or that have multiple numbers that belong to each category. So you'll notice that this count data only has one number that belongs to each category. You know, females and colorblind has one number. Males and colorblind has one number. and so on, right? That's just count data. But here, let's say we're looking at diversity across these oyster reefs and bare ground and like concrete blocks. Now, what we're doing is, let's say we have multiple plots, so we're calculating diversity across multiple plots within each of these habitat types. And so what we want to do is take the mean of each of those habitat types and compare those means. Now, so this is kind of where you would have like multiple numbers belonging to each category. So here we have 1, 2, 3, 4, 5, 6, 7 numbers belonging to each category. And that's kind of indicating to you like, okay, I'm going to have to take the mean of each category and then represent it as a bar plot with some standard error bars and whatnot. This one is a little bit more complicated. It's pretty much the same structure as this one, but instead of having just one factor, which is like habitat type, we have 2 factors. So we're looking at sugar content in corn from these different days of picking, and then light regime. So we have low light regime and high light regime, but then also, you know, 1 day after picking, 2 days after picking, 3 days after picking. And again, because we have 4 values for each of these categories, so 1 day in low has 4 different categories, 4 different samples, right? So this would indicate that we again need to take a mean of this data set and visualize it in a bar graph. All right, so numerical data is, if we just have numerical data and there's no categories involved, this is kind of what it would look like. So this would be like an example of if we're interested in like relationships or associations between 2 variables. So typically what we're interested in for like these sorts of data sets is if there's a difference between the factors, or if there's a difference between the categories. So your variable, your numerical variable is always going to be the same, because you're like looking to see if diversity is different across these different factors, right? But here we don't have factors. We actually have 2 numerical variables, and we're seeing if there's a relationship between those 2 numerical variables. So what we're doing is we're saying, okay, if we increase speed, what's going to happen to pulse? So if you increase speed on a bike, what's going to happen to your pulse? And we're seeing if there is a relationship with speed and with pulse, right? So that would be maybe represented as a scatter plot. So you can see as you increase speed, your pulse will increase as well. It wouldn't really make sense for us to make a bar graph for this, because we would take the average of speed and the average of pulse, and then what would that show us, right? So then for this one, you know, we have like, this is weight of sand crabs. And so we're seeing if the weight of sand crabs is associated with the number of eggs that they carry. And so again, it wouldn't really make sense for us to like take the mean of weight and take the mean of eggs, because that wouldn't really tell us anything. We're looking for a relationship to see if there's some association, some pattern. If we increase the weight, does that increase the number of eggs? All right. So this is just a nice table for you guys. I think these tables are actually pulled from Dr. Johnson's PowerPoints. But typically, if you have some sort of categorical data, then you're going to represent that data as a bar chart or a frequency table. So anytime you see some sort of category and there's only one numerical variable that's measured, you're going to use some sort of bar chart or frequency table. Then if you just have numerical data, I guess if it's numerical data with count, then you would do some sort of histogram. But for 2 sets of numerical data, then we would also have like a scatter plot. Okay, so here this probably looks familiar because you went through this with Dr. Johnson, but this is kind of like, okay, so what's your explanatory variable, or something that it's like not your dependent variable, not the thing that you're interested in testing, right? So for example, an example of explanatory variable might be like, if you're changing temperature to see how oxygen consumption responds, your explanatory variable is the temperature, and then your response variable, the one that's responding to the one you're changing, is your oxygen consumption, right? So if you have some sort of categorical data and if your explanatory variable is categorical and your response variable is categorical, then you're going to do a mosaic plot or a grouped bar plot. Now, if your explanatory variable is categorical and your response variable is numerical, then you're going to do a box and whisker plot or a bar plot. So these two are ones that we're going to use frequently in this lab. Now we're probably not going to use them until like the second half of the lab. We're going to get some practice with making those sorts of plots today. But these two are probably one of the most common. Actually, I'll just say like these down here are actually the main plots that we will be using for this course. And then this is what I was talking about. If we have numerical data and numerical data, oxygen consumption and temperature, then you're going to use a scatter plot. This is not something that we typically use. This is a very like uncommon data set that we see. So we're not going to go over that one today. We're just going to we're going to focus mainly on the ones that we will learn for this lab. Okay. All right, so here's a nice flow chart that I created to help you decide what kind of plot you would use. So, and these are just like all the plots that we would possibly use for this course, right? And I also want to emphasize that you guys will never be tested on making a plot. You'll never have to like make a plot for an exam and then provide me the plot and then I grade you on the plot. You'll just have to make the plot so you can describe to me correctly what's going on in the data. That's it. Okay, we'll get there when we get there. So the first question is, do we have any categorical data? And so if it's yes, then you're going to move on to this next question. And you'll notice that this whole entire half of the categorical data tree is going to be like some sort of bar plot, right? So the next question you want to ask yourself is the data count? That's like discrete. Did you actually count the numbers of each individual in each category? Or are you comparing means? So that was that one slide that I showed you where we had that count of color blindness in males and females, and then we had like the diversity in those different habitat types, right? So that was count versus calculating means. So if it is count, then you're going to use a histogram or a stacked bar plot. So that's what you see here. And it basically just kind of shows you the proportion that you see in each of those categories, right? It just kind of helps you visualize the patterns that are going on. So for example, like when we looked at that table beforehand, it was like we could look at the table and be like, okay, I guess there's more males that are colorblind than females, but it's actually just easier for us to actually visualize that in this plot. I realize that it doesn't have the color blindness labels on there, but we'll get into it. So then if we go back to this table, or sorry, this question, is the data count or are you comparing means? So if it's not count and you have multiple numbers per category, that means you're going to calculate a mean. So we're going to deal with means. So we're going to either represent that data with a bar plot. This is the most correct way to represent data that is means, because a bar plot is representing means, and these are representing the standard error around the mean. The other way we can do this is representing these box plots, which are nice because it allows you to kind of see that variability in the data. But this is actually if you're calculating a median. So if we remember from our descriptive statistics last week, the median, actually I don't even know if maybe that is, no, that's probably next week. But this is like median graphing and this is mean graphing. So you just have to be careful about that. Okay, now if we go back to our question here, do we have any categorical data? Let's say we're dealing with oxygen consumption and temperature. My answer would be no, because I just have two different numerical variables. So then the next question you want to ask yourself is one of the variables controlled for? This is something we'll kind of get into later on when we do regression and correlation. But if your variable is controlled for, then you want to do a scatter plot with a regression line, because you're going to do a regression. Regression is like kind of looking at the relationship of something, and you can really only say if there's a relationship if you actually controlled for a variable to see how the other one responds. Now, if you didn't control for a variable, let's say you just measured somebody's weight and their cholesterol content, right? You can't control for their weight, you can't control for their cholesterol content. So that's just a plain old scatter plot. All right, so this is our examples of our count data. So here we have that colorblind one. So remember, if you can look at the table, you can kind of see, like, yeah, I guess there's more females that are not colorblind, and there's like, on average, I guess, more males than females that are colorblind, right? But it's so much easier to just take a look at the graph. So here we have normal vision, carrier, and colorblind. We can actually visually see that there are more males that are colorblind than females, and there are no males that are carriers, right? So you guys remember how colorblindness works in males and females, those genetics, those two X chromosomes. And then we have the normal vision, which is like kind of half. Here's another kind of representation of this count data that we can deal with. So we have age versus the number of individuals that is within that age bin. And so we're basically like creating these bins based on age, and we're just literally looking at the number of individuals that are that age. And it kind of follows this bell curve where we see a lot of individuals that are at ages 40 to 49, and then it kind of decreases as you go out to the extremes. All right, so here we're looking at our means again. So this is our corn sugar content by days in picking and then light regime. So because we have two categories, we actually have to account for that. So we have 2 bars per one of those categories. So we have our days since picking category on the bottom, but then it's colored by light regime. So we have 2 different factors going on, but we have one numerical variable, which is the sugar content. Sugar content is represented on the y-axis. And so we can see that, okay, it looks like maybe, you know, in high light regimes, we can see an increase in sugar content, but in low light regimes, maybe it stays the same, right? Finally, we have our little diversity plot for our different categories here. So we have our oyster reef diversity, bare ground diversity, concrete block diversity. So what you could say, or based on this graph, you can, instead of like trying to look at these values, right, we can just look at this graph and say, hey, it looks like oyster reef has greater diversity than both bare ground and concrete block, right? A lot easier for us to just visualize the data with a graph than looking at the data itself. All right, so then finally we have our numerical data. So this was if you have 2 different variables that you're measuring. So here we're having that speed on the bike versus the pulse. So as you increase the bike speed, you're increasing the pulse. And for this problem, let's assume that you're like a physical therapist and you are physically increasing the speed on the bike for your patient. So if you are controlling for the speed on the bike, then you're going to do a regression and you're going to have that line on your scatter plot. It's not super important for right now, but if you're controlling for it, typically you're just going to have that regression line. This one, you're doing weight versus eggs in that sand crab one, right? So if we increase the weight of our sand crabs, does it increase the eggs? And you can't control for the weight of a sand crab, and you can't control for the number of eggs that they have. So this would technically just be a correlation. So you just kind of look to see if there's some kind of correlation going on. And it's just a plain scatter plot, no regression line. All right, so now we're going to go ahead and get into our activity for today. So you guys should already have those Excel sheets downloaded. I'm going to go ahead and just walk through. We're going to walk through how to graph 3 different graphs in Excel. And then we're going to break into our groups again and break into our groups again, and then we're going to work on some data visualization on our own. Okay. All right, so on the second tab, you have new functions. I wouldn't worry too much about this part right here. This is kind of when we get into the introduction to R. We'll see if we get there today. But I also will always provide you guys with CSVs, so you guys never have to worry about saving the CSVs as CSVs. Don't really worry about that part. But this is just kind of like new functions that we're going to learn today to be able to graph the things that we want to graph. All right, so maybe you guys already downloaded that paper that I provided you on Beachboard. If you didn't, if you want to download it now, you can. I'm just going to kind of walk you through it. and explain to you what is going on in the paper. I chose this paper because it has a lot of graphs on there, and it's a pretty, I mean, if you read through it, it seems kind of dense, but it's actually a pretty simple topic, so I'll just kind of like explain it to you as I go. Let me open up the paper really quickly. Does anyone have any questions about data visualization for me? Okay. All right, so hopefully you're all with me on your Excel sheets and you're on the first tab called Data Display Example 1. All right, so I just want to review what we're looking at here. So for this data set, we're looking at UFA concentration and oxygen consumption. So can somebody, so UFA concentration, well, let me explain what is everything that we're dealing with for the moment. Okay, so the idea here, what we're doing in this paper is we are actually, or what they did in this paper is they recorded respiration rate in 3 different cell types. And they recorded respiration rates essentially, or oxygen consumption, in a prokaryote, E. coli, a eukaryote, and then an isolated mitochondria. So the idea here is that if you think about like kind of where all of the cellular metabolism actually happens, it happens in the electron transport chain. If we can remember back to like general biology, all of that ATP generation and oxygen consumption happens at the electron transport chain. And if you can remember, the electron transport chain lives within a membrane. And if we think about membrane fluidity, if you guys remember how membrane fluidity works, we have saturated lipids and we have unsaturated lipids. So you can have these little lipids that, so these, we have like lipids that are straight like this, and then we have lipids that have a little bend in them, right? So we have these saturated lipids that are straight and unsaturated lipids that are bent. And so if you think about like butter versus, so butter versus olive oil. So butter is an example of a saturated fat because they all stack together and they can remain solid at room temperature. But olive oil is an example of unsaturated fat. It's more fluid because they don't stack together very well because of that bend in the carbon chain in their lipid, right? So the idea for this paper was we were supposed to, or what they wanted to find out is, does membrane fluidity impact the rate of oxygen consumption? So the idea here is that if you increase fluidity of your membrane, so remember all the electron transport chain is within that membrane. So if you increase the fluidity of that membrane, everything can move faster. And so you can get faster, the electron transport chain will move faster and you can get faster oxygen consumption, greater oxygen consumption, right? If you increase that membrane fluidity. All right, so what we're looking at here is these are 2 different two different variables. UFA concentration is the unsaturated fat concentration. So the greater the number, the more fluid the membrane is, right? And then this is oxygen consumption. So the greater the number, the higher oxygen consumption. So is this, can I get maybe some answers here in the chat or something? Is this do we have any categorical data here, or is it strictly just numerical? What do you guys think? Awesome. A lot of you are correct. It is just numerical, right? We have no categories. We have 2 different numerical variables that we're looking at. We have the unsaturated lipid concentration. So that's like the membrane fluidity. And then we have the oxygen consumption. So what kind of graph are we going to use? What do you guys think? Are we going to use a bar plot or are we going to use a scatter plot, basically? Awesome. We're going to use a scatter plot. Okay. So if you want to follow along with me, I'm going to show you guys how to create a scatter plot in Excel. If you already know how to do this, just bear with me. Some people are more Excel savvy than others. So what you're going to do is you're going to highlight. So you're going to find that big black, the big white crosshairs, and you're going to click and highlight your entire data set. All right, so then we're going to go up to your taskbar up here where it says Insert. You're going to click on Insert. All right, and then there's going to be different categories in your insert box here. We're going to go to the category called Charts. All right, so we want to do a scatter plot. So what you want to do, you can do recommended charts, and that will give you there will be a scatter plot in there. But we want to specifically tell it to do a scatter plot. So you find the plot that looks like a scatter plot. That's this one right here. And when you hover your mouse over it, it says Insert Scatter. So I'm going to click on that. And then we're just going to do a plain scatter plot. Okay, so you guys should all have your scatter plot there. Now, if we did not control, let's say we didn't control for either variable, and we just recorded the membrane fluidity and the oxygen consumption, is this plot okay? If we did not control for anything. What do you guys think? Is the plot okay? Awesome, I agree with you. Yes, the plot is fine. If you do not control for anything, then you're just going to do a correlation, right? You don't want to have that regression line on there. Now, one more thing I wanted to go over is how do we actually title our axes? So if you go over, there should be a plus sign right next to your plot. So you can click on that plus sign, and then there's a bunch of chart elements that show up. We want to click axis titles. Your axis title text boxes show up. So what we want to put on the x-axis is our UFA concentration. Whoops, that's not UFA. So we have UFA concentration on our x-axis. And then we have O2 consumption on our y-axis. So again, to get those axis titles, you do the plus and then go to axis titles. If you don't see them, right click on your plot, and then I think it is format chart area, I believe. And you can add axis titles that way. If you format chart area, then you get a whole pop-up over here. Let's see. Oh, text options maybe? Nope, I'll have to figure out how to do that without that, I guess.
You can also double click on the chart, and at the top left corner it should say add chart element, and that's
awesome.
where, yeah.
Okay. All right, so now let's go ahead and assume that we actually controlled for membrane fluidity. So there is a way that you can control for membrane fluidity. There's this enzyme that will actually basically transform the saturated lipids in the membrane to unsaturated lipids in the membrane. So it will make the membrane more fluid. And so you can basically add this gene into your cell to transform those saturated lipids to the unsaturated lipids to make it more fluid. So let's go ahead, and that's kind of like what the paper talks about. So let's go ahead and assume that we controlled for membrane fluidity. So if we controlled for membrane fluidity, what do we want to put on our graph?
Regression line.
Yep, we want to put a regression line on there. So it's going to be another element that we're going to add onto our plot. So if you have that plus sign, or you can double click and go to add chart element. So plus sign, and then we want to add a trend line. And then you want to click the trend line that you want to add. Whenever we do regressions, it's always going to be a linear regression. So we're going to click linear. And there's your line. All right, so I'm going to go ahead and show you those graphs in here that represent that. So basically what we see here, this is in our prokaryote. So what we see is that as we increase this on the x-axis for all of these plots, as we increase our unsaturated fat, then we increase our respiration rate as well. And then this plot over here is showing that example where we actually controlled for membrane fluidity. So this is that FAB-H2 gene that I was talking about that actually transforms those the saturated fats to unsaturated fats. And so you can see that when you include that enzyme in there, it actually does increase the respiration rate because you're increasing the fluidity. So there's some good representations of those scatter plots. We also have some similar scatter plots in here for our eukaryote. So here we have, as you increase your unsaturated fatty acid in your eukaryote, and you're also going to increase your respiration rate. All right, so let's go ahead and move on to the next example here. So we have data display example 2. All right, so what we're looking at here is oxygen consumption averages across 3 different cell types: eukaryote, prokaryote, and an isolated mitochondria. Remember, that's what the paper was studying, those 3 different cell types. And so we're going to compare oxygen consumption rates across all 3 of these different cell types. So do we have just numerical data, or do we have some categorical data here? What do you guys think? Awesome. Yes. So we do have both, but the key here is that we do have categorical data. So if we have categorical data, what kind of plot are we going to try to move towards? Bar plot, or yes, bar plot. Awesome. All right, and this is also kind of a hint. So if you get a data set that has a mean SDSE, that kind of is a hint that you're going to do a bar plot, because a bar plot is representing means. All right, so what we have to do first is we actually have to, if we want to create a bar plot in Excel, we have to calculate the summary statistics. And those summary statistics for that bar plot include the mean and the standard error, because you have to plot the standard error, and then you have, or sorry, plot the mean, and then you have to add these standard error bars onto your plot. So what we're going to do first is we're going to go ahead and calculate our means. So do you guys remember how to calculate a mean from last class? Actually, I think we learned it last class, right? Yes, average. So every time we tell Excel we're going to calculate something, you always start the cell with an equal sign, right? I'm going to zoom in here so we can see better. Oops, okay, hold on. Okay, so I'm going to do equals and then average. Your average function should pop up. You're going to double click that. And then we're going to select all of the numbers, within the eukaryote column. Okay, now remember that we don't have to do that for every column. We can actually just do the drag and drop, but let's go ahead and calculate the rest so we can just drag everything to across the rest of the columns. So let's go ahead and calculate standard deviation. So we're going to do, and the reason we're calculating standard deviation is, and we haven't quite learned this yet, I think we're learning it next week, but the reason we want to calculate standard deviation is because to calculate standard error, you need to calculate standard deviation. And so the standard error is standard deviation divided by square root of the numbers, or the n, basically. So we're going to go ahead and calculate standard deviation. So the function for standard deviation is stdev.s. So there's a .p, but we want to choose the .s, and that's because .s stands for sample, .p stands for population. We never really know the entire population of something, so we hardly ever use the .p function. We always typically use the .s function. So we're going to use standard stdev.s, and then you're going to select the same numbers. And that should be your function, stdev.s. All right, so there's our standard deviation. And now what we want to do, let's see, why can't we just use stdev? You might be able to just use stdev, but I'm not sure if you'll get the same exact number. Will you get the same exact number when you calculate it? We try to stay like consistent with all of the functions so that you guys will get the right answers on the exams. Okay. I'll have to look into see like what that difference is in those calculations. Okay, so then we're going to do standard error. So standard error is standard deviation divided by the square root of your n, your sample size, basically. And so we're first going to put an equal sign, and we're going to start creating our little calculations. So we're going to do standard deviation on the top of the numerator. So we're going to select our standard deviation. Then we're going to put a divided by. And then remember, it's the square root of the sample size. So we're going to do SQRT for square root. SQRT. All right. Now, you guys can actually go, and if you want, you can calculate or you can count each one of these values, or you can have Excel do it by using the function count. So I'm going to use the function count and show you how that's done. So we're going to do square root, and then we're going to tell Excel to count the number of rows in that column. So we're going to do count. So you should have SQRT parentheses count. And then when you highlight all of this, Excel is basically going to count all the numbers in that row. So you don't have to do it yourself. Now notice that we have two different colored parentheses. That's because it's just letting us know like which set of parentheses is which. So we know that we've closed this red parentheses, but we also need to close the black parenthesis to complete the equation. So I'm going to close the black parenthesis as well. All right, so you can go ahead and press enter once you have that. And there is our standard error, 27. All right, now remember, you don't have to do this for each of those cells. What we can do now is we can just highlight all three of those cells. I highlighted them using the white crosshair. So I highlighted all of those cells. And now what I want to do is I want to find that black crosshair on the bottom right-hand side of my Excel sheet. Or sorry, my green highlighted box here, right there. And I'm going to click, hold, and drag it across. So it calculates everything else for me. All right. So once we're here, now we can actually graph our data. So we can graph our means. So what we're going to do is we first just want to select the mean so we can get the bar plot. Then what we're going to do is add on the standard error bars. So let's go ahead and first plot the means. So first you're going to highlight your categories and your means. We're going to go up and we're going to click on insert. You can do recommended charts, or you can do, or just find the bar plot, or the bar plot icon. You're going to click on that, and then I typically just use this first one here. Okay, so there is our plot. Those are our means. Now what we want to do to make a correct bar plot is we want to add on those standard error bars. And this is a little bit of there's a lot of steps to this part, so just try to, I'll go slowly. So what we want to do is we want to add on our error bars to our plot. So one way you could do that is by adding chart elements. So either with that plus sign or double clicking and then add chart elements. So we can go click here, and then we want to add on error bars, but you don't want to just click error bars because you want to actually customize what the error bars should be. So we're going to go to this little sidebar here, and we're going to go to more options because we want to actually tell Excel what standard error we want to use. So we're going to go to more options. And you should get a little pop-up on the side here, starting with format error bars. All right. Now what we want to do is we want to, let's just keep all these things the same. You want it to be both. So you want the tails to be on both ends, and you want there to be a cap. Those are just generic error bars. We'll keep it that way. And then let's, instead of having fixed value, we don't want fixed value. We want custom. So we're going to tell Excel which error bars we want to use. So you can click custom, and then you want to click specify value. So I'm going to click specify value. And now this is where you want to enter in all of your error bars. So this is the plus error bars and the minus error bars. We're going to enter in the same numbers for both. So what we're going to do is, this is highlighted right now. So you can just delete that one. And then we're going to select all three of our standard errors, and it will plot it in order. The next thing you want to do once you have the positive one in there is you want to go down to the negative. Oh, hold on. Let me try this again. Okay. For some reason, it's not letting me do the negative. Let's see. Well, typically you want to go down, click the negative, delete that, and then also put the negative in there. But I'm going to press OK, and we'll see what happens. Specify value, negative. Okay, I guess I just have to do it. Something's wrong with my Excel for now. So you should be able to put both the positive and the negative in there. So you'll highlight all of those standard errors for both the positive and the negative. So it's the same numbers. We should get the same sort of error bars up, or like plus and minus of our means. And we have our complete chart there. And then again, you can add your chart title and your chart or your axis titles. So I might put my axis titles on here, and I might put cell type. And then over here, I'll put oxygen consumption.
For the negative error value, are you supposed to put a negative sign?
Nope, you don't have to put a negative sign. You just select the same ones, and it'll just put it like below.
Okay.
Did you get an error?
Because mine doesn't look the same as yours.
Let's
I don't know
see.
if I did it wrong.
When are you using a Mac? Do you have a Mac or no?
Yeah, I do.
You do.
Because
Okay.
like my bars are behind the graph, or like my standard error lines are behind the bar. Or I don't know if I did it wrong.
Like, do you see the top one, but you don't see the bottom one?
Yeah. Oh wait, I figured it out. I'm sorry.
Okay. It's probably just because you didn't put the bottom one on there. Yeah.
Yeah, okay.
All right, cool. All right, so what kind of pattern, just visualizing the data, what kind of pattern do you guys see? What do we see? What can we conclude, or what can we kind of describe from this graph? Can we conclude that all of them have the same oxygen consumption? Yeah, I would say no. So what we see is that eukaryotes have greater oxygen consumption than prokaryotes and isolated mitochondria. And that makes sense because eukaryotes are larger than prokaryotes and isolated mitochondria. Prokaryotes and isolated mitochondria have similar oxygen consumption rates. And that makes sense because they are of similar size. And if we also think about kind of the evolutionary pattern that these cells took, if you guys remember from general biology, that symbiosis theory, it's thought that mitochondria were once on their own. They were their own prokaryotes. And so that's why they're very similar to prokaryotes, because they were once prokaryotes, but just engulfed by another larger cell that became a eukaryote. All right, so that's also stated in the paper, just comparing them and saying, okay, you know, eukaryotes have larger oxygen consumption, and prokaryotes and isolated mitochondria have similar, and going back to that symbiosis theory. Okay, let's go ahead and do one more together. Let's go to data display example 3. Okay. So we have the 3 different cell types: isolated mitochondria, prokaryote, and eukaryote. And then we have 4 different categories of lipid membrane types. So we have cholesterol, hapanoids, glycolipids, and palmitic acid. So these are all just types of lipids that exist in the membrane. Now, let's go ahead and go through the, we'll just graph the plot first. So we only have one number belonging to each category. And so what kind of data are we dealing with here? Are we measuring something, you think, or are we counting something? If we have one number belonging to each category, what might that imply? Yeah, so this would be count data. So this is saying that we found 5 cells that, or sorry, 5 isolated mitochondria that have cholesterol, 0 prokaryotes that have cholesterol, 67 eukaryotes that have cholesterol, 51 isolated mitochondria that have hapanoids, and so on, right? 0 eukaryotes that have hapanoids. And we'll talk about this pattern when we get there. So let's go ahead and visualize the data so we can see what's going on. So what we want to do, this is super simple. We just kind of want to show proportions, and there's multiple ways you can kind of show this sort of data, and I'll show you those different ways. But how maybe I like to show this data is if, let's go ahead and just highlight the entire table, except for cell type and lipid membrane. We're going to go to insert. And then let's go ahead and go to our stacked bar plot. Okay, I'm going to make this a little bigger. So what this is showing us, it's literally just the counts that exist in each of the categories. So what we see is that we have cholesterol on the x-axis, or cholesterol, hapanoids, glycolipids, and palmitic acid on the x-axis. And then it's colored by isolated mitochondria, prokaryote, and eukaryote. Now, let's say that maybe we don't think this is the most intuitive way to graph this data. Maybe we want to switch which ones are on the x-axis and which ones are the colors. So let's go up to this data portion right here, and let's say switch rows and columns. I'm going to click that. So I personally like this a lot better. So now we have these cell types on the x-axis, and it's now colored by the different types of lipids. So what sorts of patterns can we notice here? Anyone notice any patterns? For starters, they all have palmitic acid. They all have glycolipids. Cool. Any other patterns that you guys notice or lack of?
How do we read this graph?
So basically this is on the x-axis, it's just the count. It's literally just the count. And so what we have to, and sometimes we don't love these types of graphs because it's basically like you have to kind of do math to figure out what's going on to see the actual count. But it's really just representing like sorts of like a proportion almost. So we can visually see that there are more, or prokaryotes have more haploids, and isolated mitochondria have more haploids than eukaryotes, which have no haploids, right? Eukaryotes have way more cholesterol than prokaryotes and isolated mitochondria, because we can see that there's no blue in prokaryotes and very little blue in isolated mitochondria. Does that make sense? And then this is just kind of like representing the numbers that have glycolipids. So it's like they kind of all have similar glycolipids and they all have similar palmitic acids.
So like if I'm trying to read the haploids for isolated mitochondria, it's 51, right? And so I wouldn't think of it as like a continuation of the cholesterol. Would I? Like,
oh,
how would I
no,
read it
yeah.
specifically?
So it's like added on, right? So this is like, so for example, if we look at this isolated mitochondria one, we have 5 that are cholesterol. And then this is just starting, like this entire box represents 51. It's not adding on. Good question.
Oh, okay. Thank you. That makes more sense.
Sorry, can you show us how to switch the columns?
So up here, and so we're on like chart design tab. So you have to click on your chart. So click on your chart, and then in chart design, there'll be a little box up here that says switch row/column, or maybe an icon that has little arrows like that. All right, so you guys basically told me, yes, and then we kind of went over it, that there are haploids in isolated mitochondria and prokaryotes, but there are no haploids in eukaryotes. There's cholesterol in eukaryotes, and there's no cholesterol in prokaryotes and isolated mitochondria. And if we think about cholesterol is kind of like, it's an element that attributes to that membrane fluidity. And so evolutionarily, eukaryotes do have cholesterol, but prokaryotes do not have cholesterol. So what this is also showing us is that similarity between mitochondria and prokaryotes in their membrane kind of construction almost, because they both have haploids, which is more of a prokaryotic lipid that is involved in their membranes, versus eukaryotes just have cholesterol. They don't have haploids. And then I'll just quickly show you like a couple different graphs that we can use. So instead of using the actual count, we can also represent this as proportions. So if we choose the proportion one, now we can look at it in terms of percentages. And if I switch this, now this is in terms of percentages and it's actual proportions. So you'll notice that it's all the same height. Versus this is like actual counts. So we obviously have more eukaryotes that we counted overall versus prokaryotes and isolated mitochondria. But if we put it in proportion, sometimes this is better to visualize the data. The other way that we can also represent this is just by doing another bar plot. So if we did this bar plot over here, now we can see, oh, it's gone now, but now we can see that we have like no cholesterol here, lots of haploids, glycolipids, and then so on, right? So that's kind of another way we can visualize this data. So for this kind of data, you can do multiple things. This one, you wouldn't add error bars because it's just count, right? All right, any questions for me? All right, glad you guys are enjoying this. Okay. All right, so let's go ahead. So I believe, how did you do the last graph? Okay, I basically just, so let's just insert this here. So I did insert, and then I went back to that bar plot, and I actually used the same bar plot that we used in the other example, but then I also just switched the rows and columns, so it seems more intuitive for me to read. I guess it looks a little different this time. Yep. Okay. All right, so what I want you guys to do now is I'm gonna stop sharing my screen. So we're gonna do some group work now. So what I'd like you guys to do, first you're gonna turn your cameras on, and you guys should all know which group you belong to, and I believe I do have 7 groups. Yep. So you'll see on the bottom of your Excel sheet, you see different tabs. There's group 1, group 2, group 3, group 4, and so on, right? So whichever group you are in, you're going to work on that tab. So group 1 is going to work on the group 1 tab. And then group 2 is going to work on the group 2 tab. Group 3 is going to work on the group 3 tab. And basically what you guys are going to do is you're going to read how that data was collected. And then you're going to create a plot. And then once you guys are finished, we'll come back together as a class, and I'll have somebody from your group share their screen. And each one of the groups will share their screen and talk about what they did for that plot and why they chose that plot. Oh yeah, you guys can take it. Let's take a break. We'll take a break. We'll come back at 10:40. How about that? 5 minutes? Get some coffee and snacks?
Thank you.
Yeah. Yeah, please remind me about breaks. I usually am good about taking breaks, but so far, lately I've not been. All right, we'll come back at 10:40.